Cell membrane.
The cell membrane is the thin, flexible boundary that surrounds an animal cell and separates its interior from the outside environment. It is mainly made of a double layer of phospholipids, with proteins embedded throughout it. Rather than acting like a solid wall, the membrane is selectively permeable, allowing some substances to pass while restricting others. This lets the cell control nutrients, water, ions, and waste products and maintain a stable internal environment. Cytoplasm. The cytoplasm is the entire region between the cell membrane and the nucleus, and it includes the cytosol together with the structure suspended within it. The cytosol is mostly water, containing ions, proteins, sugars, and other dissolved molecules. It provides the environment where many important chemical reactions occur and allows substances to move between different parts of the cell. Nucleus. The nucleus is the main storage site for an animal cell's genetic material. It is enclosed by a double membrane called the nuclear envelope, which contains pores that regulate movement between the nucleus and cytoplasm. Inside, DNA is organized with proteins into chromatin. The nucleus uses this genetic information to regulate which proteins the cell produces, influencing processes such as growth, metabolism, repair, material repair, and cell division.
Nucleolus. The nucleolus is a dense region located inside the nucleus, rather than a separate membrane-bound organelle. Its primary job is producing ribosomal RNA and assembling the components that will become ribosomes. Several nucleolus regions can sometimes be present within a nucleus, depending on the cell. Because ribosomes are essential for protein production, the nucleolus is closely connected to the cell's ability to manufacture proteins. Ribosomes. Ribosomes are microscopic molecular machines that build proteins. They consist of ribosomal RNA and proteins and work by reading messenger RNA, which carries instructions copied from DNA. Ribosomes can float freely in the cytoplasm or attach to the rough endoplasmic reticulum. Free ribosomes generally make proteins used within the cell, while ribosomes attached to the rough endoplasmic reticulum make many proteins destined for membranes, organelles, or secretion outside the cell.
Rough endoplasmic reticulum. The rough endoplasmic reticulum is an extensive network of interconnected membranes covered with ribosomes. It is called rough because those ribosomes give its surface a rough appearance under a microscope. Proteins made on its attached ribosomes enter the endoplasmic reticulum, where they can begin folding and undergo early processing. The rough endoplasmic reticulum also produces membranes and packages proteins into transport vesicles that travel toward other parts of the cell, particularly the Golgi apparatus.
Smooth endoplasmic reticulum. The smooth endoplasmic reticulum is another part of the endoplasmic reticulum network, but it lacks ribosomes on its surface. It is involved in producing lipids, including molecules used to build cellular membranes. It also participates in detoxification and chemical processing, particularly in certain cells, such as liver cells. In muscle cells, a specialized form called the sarcoplasmic reticulum stores and releases calcium ions, which are essential for muscle contraction. Golgi apparatus. The Golgi apparatus is a series of flattened membrane sacs that acts as an important processing, sorting, and shipping center. Proteins and lipids arriving from the endoplasmic reticulum are modified as they move through the Golgi. The Golgi then sorts these molecules and packages them into vesicles according to their destinations. Some materials are sent to the cell membrane, some to lysosomes, and others remain within the cell. Mitochondria. Mitochondria are membrane-bound organelles that produce much of the ATP used to power cellular activities. They break down molecules derived from food and use the released energy to drive cellular respiration. Each mitochondrion has an outer membrane and a highly folded inner membrane, with the folds called cristae. These folds provide additional surface area for reactions involved in ATP production. Mitochondria also contain their own small amount of DNA and ribosomes, reflecting their evolutionary history.
Lysosomes. Lysosomes are membrane-bound compartments containing enzymes that break down biological materials. They can digest damaged cell components, certain particles brought into the cell, and other materials that are no longer needed. Their enzymes work best in the acidic environment maintained inside the lysosome. After materials are broken down, useful components can be returned to the cell for reuse, making lysosomes an important part of the cell's recycling system. Peroxisomes. Peroxisomes are small membrane-bound organelles that carry out several types of chemical reactions. They help break down certain fatty acids and participate in the processing of potentially harmful molecules. Some of their reactions produce hydrogen peroxide, which can damage cells if it accumulates. Peroxisomes contain enzymes, such as catalase, that help break hydrogen peroxide down into less harmful substances. Their exact functions vary between different types of cells. Vesicles. Vesicles are small membrane-bound sacs that transport, store, or deliver substances within the cell. They allow materials to move between organelles without those organelles having to directly connect with one another. For example, transport vesicles can carry proteins from the endoplasmic reticulum to the Golgi apparatus. Other vesicles can carry materials from the Golgi to the cell membrane, lysosomes, or other destinations.
Centrosome. The centrosome is a region of the animal cell that organizes microtubules, one of the three major components of the cytoskeleton. It is generally located near the nucleus and contains a pair of centrioles surrounded by protein-rich material. By organizing microtubules, the centrosome helps establish the cell's internal structure and contributes to the movement and positioning of cellular components. It becomes especially important during cell division. Centrioles. Centrioles are cylindrical structures made primarily from microtubules and are usually found as a pair within the centrosome. They are arranged roughly perpendicular to one another and help organize microtubule structures. During cell division, centrosomes containing centrioles help establish the spindle system that separates duplicated chromosomes. Centrioles can also serve as basal bodies, which act as organizing centers for the formation of cilia and flagella. Cilia. Cilia are short, slender projections extending from the surface of certain animal cells. They contain organized bundles of microtubules and can beat in coordinated patterns. In cells lining the respiratory tract, for example, coordinated cilia movement helps move mucus and trapped particles along the surface. Cilia can also perform sensory functions in some specialized cells. Their structure allows them to convert energy from molecular motors into controlled movement.
Flagellum. A flagellum is a long, whip-like cellular projection that can produce movement. In humans, the best-known example is the flagellum of a sperm cell, where its rhythmic motion helps propel the cell. Like cilia, an animal cell flagellum contains a specialized arrangement of microtubules and molecular motor proteins. Although cilia and flagella share many structural features, a typical flagellum is much longer and is primarily associated with propulsion. Microvilli. Microvilli are tiny finger-like extensions of the cell membrane that greatly increase a cell's surface area. They are especially abundant on cells specialized for absorption, such as cells lining the small intestine. Increasing surface area gives the cell more membrane space for transport proteins and other molecules involved in absorbing nutrients. Unlike cilia, microvilli are not designed to beat back and forth. Their main purpose is to increase surface area and support absorption. Cytoskeleton. The cytoskeleton is an interconnected network of protein fibers that gives an animal cell structure and organization. It consists mainly of microfilaments, intermediate filaments, and microtubules, each with different properties and functions. Microfilaments help with cell shape and movement. Intermediate filaments provide mechanical strength, and microtubules act as tracks for intracellular transport and help organize cell division. Together, these systems allow the cell to maintain its shape, move materials, change its structure, and coordinate complex activities.
The cell membrane controls what enters and leaves, while the cytoplasm provides the environment for many cellular reactions. The nucleus stores DNA, and the nucleolus helps produce ribosomes, which build proteins. The rough and smooth endoplasmic reticulum make and process proteins and lipids, while the Golgi apparatus modifies, sorts, and packages them. Mitochondria produce usable energy, lysosomes break down and recycle materials, peroxisomes process fatty acids and harmful molecules, and vesicles transport substances around the cell. The centrosome and centrioles organize microtubules, while cilia and flagella enable movement of materials across the cell surface. Microvilli increase surface area for absorption, and the cytoskeleton provides structure, organization, transport, and movement. Together, these parts work as an interconnected system that allows the animal cell to survive, grow, respond, and reproduce.